All right, so this lecture is very short on detail. The reason for this is that the immunology of the skin or the skin associated lymphoid tissue, which we call SALT, is an area of very active research for the past 30 years. The problem is that skin immunology or the skin is very difficult to study, to do research on, because not everybody is willing to have bits of their skin punched out to do experiments on. So most of the data that we have with regards to the system comes from experimental mice. And the problem is that mice anatomically are very different to humans, that because they have so many hairs, they have all these hair follicles. So what we understand about skin immunology from the mice doesn't necessarily transpose to the human system. But nevertheless, we managed to get some details out of all of this work. A lot of the themes or the complexities of skin immunology follows this whole system of barrier immunology, the immunology of the respiratory tract, the immunology of the GI tract. You already had a lecture on the mucosal immune system last year in your PhD block. So there are similar themes that evolve from what happens with the GI tract and the skin. One of the problems with this barrier immunology is that in these barriers, the epithelial cells are exposed to both commensal bacteria or microbiota and potentially harmful bacteria. And so the immune system needs to discriminate against what is harmful and what is beneficial. And we're still trying to work out how it does this. The other thing is that if there is a pathogenic invasion, the response to that needs to be tightly regulated to make sure there isn't massive collateral damage to healthy cells. So having said that, the objective of this lecture is to talk about the components of the immune surveillance within the skin, and then to describe a new concept that I never told you about in the POD block, and that is the fact that parenchymal cells themselves, such as the epithelial cells, have things like PRRs, so they can behave or function similar to the innate immune system, and so then activate the adaptive immune system. So what we think of as classically immune cells isn't exclusively limited to macrophages, lymphocytes, etc. That parenchymal cells themselves also have an immune type function. Then to talk about the influence of the microbiota in educating or training the system, the immune system, in terms of tolerance versus inflammatory responses. And then another aspect of skin immunology is the fact that within the skin, you have many openings, such as hair follicles and sweat glands. And these are openings which are exposed to the outside environment, whereby you can get microbes coming in. And so within these openings, the regulation of the immune system is even more tightly regulated to either initiate tolerance to any microbes that colonize the hair follicles or to attack whatever comes in. And then finally, I will give two examples or three examples of skin diseases whereby there's immunopathology as a result of hypersensitivity reactions against particular situations. So the skin is regarded as the largest organ in the body because it covers such a big surface area. And because of that, the immune cells that are surveying the skin make up the largest components of lymphocytes that you have in the body compared to the circulating lymphocytes that pass through the thymus and go through the lymphatics and the bloodstream and so on. The So in the skin, as you know by now, the outermost layer of the skin is the stratum corneum, composed of these dead cells which form this hard physical barrier which keeps all the moisture inside the body and also prevents moisture outside the body getting in. Underlying this is the stratum granulosum, consisting of these live epithelial cells. And within this, within the cells here, these provide large amounts of the chemical and biological molecules for protecting and preventing colonization of pathogenic outside bacteria in the form of sebum, which blocks or attacks lipids, and predominantly the so-called antimicrobial peptides, which I touched about during the POD block, but didn't talk much about. These antimicrobial peptides are very important compounds produced by parenchymal cells as well as immune cells to kill any pathogenic bacteria. And then within the stratum spinosum are three major cells: the Langerhans cells, gamma delta T lymphocytes, and resident memory T cells. So the Langerhans cells are the only myeloid cells in this layer. And for a long time, there was a lot of controversy about these cells, about whether they were actually dendritic cells or whether they were macrophage or derived from macrophages. It's now accepted that they are myeloid-derived cells. So in other words, they originate from the same precursors that monocyte macrophages originate from. But nevertheless, they have dendritic cell function in that they're important for antigen capture, presentation to T helper cells. They are characterized, I think Dr. Brass told you about Langerhans cells? No. So they're important sentinel cells within this layer of the skin, and they're characterized by the presence of these Langerhans and Burbeck granules and high expression of class 2 MAC, which relates to their antigen presentation function. Within this region, there is a large network of connective tissue. And this connective tissue serves like cables whereby these Langerhans cells, even though they're immobilized in this area, as soon as they see antigen and take up antigen, they will be activated and change their chemokine receptors, which allows them to move along this connective tissue to the nearby draining lymph nodes, where they will then present the processed antigen to the circulating naive T cells and activate them just as you've learned before, normal way. The gamma delta T cells I touched briefly on last year, but I didn't say much about them. If you remember, 95% of T cells have a T cell receptor composed of two proteins, an alpha chain and a beta chain. These T cell receptors recognize predominantly protein peptide antigens presented on class 1 or class 2 MHC. The gamma delta T cell receptor is composed of a gamma chain and a delta chain. These cells, the T cell receptor has a much less diverse recognition compared to the alpha beta T cell receptors. And the antigen that they recognize are predominantly lipid molecules. So they are more directed against membrane or capsule antigens of bacteria. The other thing is that these gamma delta T cells are more active or more associated with barrier functions like the gut, respiratory tract, or the skin, as opposed to circulating through the lymphatics and the bloodstream. But another thing about these cells is that they seem to have completely opposite effects. One, that they can directly kill virus-infected or bacterial cells, but the other is that they can produce cytokines to repair and stimulate proliferation of damaged epithelial cells to maintain the integrity of this barrier. Then the third component of this layer are these resident memory T cells, which are still a relatively new discovery. But it's now recognized that the majority of T cells in the body are actually composed of these resident memory T cells. And they are scattered throughout the skin and mucosal tissue. There are many CD8 cells, although we do find some CD4 cells, and they are important for protection against previous skin infections like measles, herpes viruses, and so on, such that on secondary challenge with or secondary infection with these viruses, these memory cells can immediately go into effect and block or kill infected cells. They are characterized by the surface marker CD69, and CD69 is an important surface marker for all activated immune cells, not just these resident memory T cells, but T cells, B cells, and natural killer cells. So they're important biomarker for seeing activated immune cells. Another thing about these resident memory T cells is that they require the cytokine interleukin 7, which if you remember from last year, interleukin 7 is an important growth factor for both memory cells as well as immature T cells in the thymus, the thymocytes. Because of the effectiveness of these resident memory T cells in attacking subsequent challenge with viruses, a lot of research in trying to develop new vaccines has gone into trying to get T cell vaccines. So not just to activate B cells to make protective antibodies, but to make vaccines that can induce this T cell memory response. Because from the COVID pandemic, we recognize that the cell mediated immune response is also a very important mechanism for protection against virus diseases. It's not just all about antibodies. Underneath the epidermis, within the underlying tissue, we find all the normal cells that you've learned about in the basics of immunology, namely the innate immune cells in the form of dendritic cells, macrophages, neutrophils, and also the T helper cells and innate lymphoid cells, and then two novel cells, which I might have mentioned, namely natural killer T cells. So these cells have both NK receptors and a T cell receptor, but they don't express CD4 or CD8. So that means that we don't really understand this cell has a T cell receptor, but it's not restricted by MHC. So whether this is a different type of recognition compared to a conventional T cell receptor. But having a natural killer cell receptor also means that it recognizes the absence of MHC expression on the target cell. What we do know about these cells is that they're important producer of cytokines, and their receptors recognize mainly lipid types of antigens. So again, directed against the lipid envelope or lipid membrane of bacterial cells. Then a subset of conventional professional antigen presenting cells, the dendritic cells. Another subset of these dendritic cells is these so-called plasmacytoid dendritic cells. These are dendritic cells with lymphoid function, T helper cell function, in that even though they can take up and process antigen, so they have antigen presentation function, their main role is to produce this type 1 interferon, which, again, although I said previously that type 1 interferons are predominantly antiviral, we now recognize that interferon alpha and interferon beta are also important activators of the adaptive immune response and inflammation. So they're important source of as a pro-inflammatory cytokine producer. Then, just to jog your memories, I said that for the innate immune response, the recognition receptors on these macrophages, dendritic cells, neutrophils, are the PRRs, pattern recognition receptor molecules. And they recognize MAMPs, microbial associated patterns, and DAMPs, which arise from sterile inflammation when you get damage to normal tissue or cells and you release these intracellular cells. These are called DAMPs. But for some reason, the literature also calls these DAMPs alarmins, which seems to be a lot more descriptive name, because when you release these intracellular contents or DAMPs, they cause alarm and then activate the nearby healthy cells to generate inflammation. One of the most important alarmins in this context of sterile inflammation is this protein, high mobility group box 1, which is a histone-associated protein. So you see, you get the release of that. Healthy adjacent cells recognize this as cell damage, and then will stimulate the inflammatory response. And then a new concept that we now recognize is that keratinocytes or parenchymal cells themselves express PRRs. So it means that the parenchymal cells, acting just like the macrophages, dendritic cells, can also pick up PAMPs, alarmins, DAMPs, and then produce your common inflammatory cytokines, IL-1, TNF-alpha, IL-6. They can also recognize these DAMPs through nod-like receptors and form an inflammasome to then generate an active IL-1 proenzyme, just like I told you about that macrophages do. So it now seems then that these parenchymal cells also act like sensors, seeing an invasion of microbes or cell damage. They will then, so we have another step where once they are activated, they can activate the innate immune response, which can then also, both of them can activate and bring in the adaptive immune response. Not only that, we also recognize, and I told you about this in the neuroimmunology lecture last year, that the skin is innervated by sensory channels from the central nervous system. So any damage, whether it's through activation of pain receptors or inflammatory responses happening in the skin, will alert the sensory neurons to send out signals to the immune system, servicing the skin to stimulate and activate cytokine production, lymphocyte proliferation, and so on. So it's all one major network that's going on here. Within these dendritic cells, when you have invasion in the skin and the activation of these inflammatory responses, and the dendritic cells getting signals from these inflammatory cytokines, as well as activation as a result of PRRs being engaged. These dendritic cells will then change their expression of particular chemokine receptor, which allows them to work their way through the connective tissue to the nearby draining lymph nodes. And then within the draining lymph nodes, they can present processed antigen to the naive T cells that are circulating from one lymph node to another. Once that naive T cell has seen that particular antigen, the lymph node, the naive T cell becomes an effector T cell, and also expresses another pattern of chemokine receptors, particularly in the form of this cutaneous leukocyte antigen. So this is a chemokine receptor, which then drives the circulating T cell not to go from one lymph node to another, but home directly back to the skin where that invasion occurs, to then generate the cell-mediated immune response. And then with regard to the skin appendages, like the hair follicles, the sweat glands, These are openings whereby there is invasion of outside microbes into these areas. But now, in addition to the potentially pathogenic microbes coming into there, you also have beneficial microbes that are part of the microbiota, like Staphylococcus epidermidis. And so here the immune system needs to be much more strictly regulated into attacking harmful microbes, but not too strongly as to cause accessory damage to healthy tissues, but also to be tolerant to the normal microflora that are in there. So here you have then a very strong, stringent balance between what is to be attacked in the form of an inflammatory response and what is to be tolerized not to react to. And any thing that is harmful, that is an inflammatory response, that inflammatory response needs to be tightly regulated as not to overblow the system and then cause hair loss as a result of attack on the hair follicles and so on. And also, in addition to the inflammatory response, it also, if once that inflammatory response has been generated, you also need anti-inflammatory cytokines in the form of IL-10 and TGF-beta to block this ongoing inflammatory response and also to repair the damaged tissue that's going on there. A lot of this is mediated by regulatory T cells, which are the major source of IL-10 and TGF-beta. So in addition to this active suppression of the immune response as a result of IL-10 and TGF-beta production by the regulatory T cells, the parenchymal cells themselves also have mechanisms to prevent active attack on them by these inflammatory infiltrates. And this is in the form of the expression of surface molecules like Fas ligand and PD-1 ligand, such that when these inflammatory immune cells start to try to attack these normal parenchymal cells, the attacking lymphocytes themselves will be suppressed or induced to die by apoptosis. So this is one way of these normal cells protecting themselves from immune-mediated damage. So the skin microbiota, just like in the GI tract, these skin microbiota, particularly Staph epidermidis, but in other types of species of the normal microflora on the skin, have an important role in educating the immune system as to what is beneficial and not to be attacked. And also, these commensal bacteria are also important for stimulating the immune cells to produce these antimicrobial peptides for killing new invaders, potentially more harmful bacteria that are trying to colonize the skin. But they, in addition to educating the immune cells to attack the harmful bacteria, they are also important for inducing the T cells to develop tolerance to tell the T cells, don't kill me, I'm good for you, but kill him because he's going to colonize you and cause pathology. But these commensal bacteria, in addition to inducing tolerance and also the production of these antimicrobial peptides, they can also, through inducing cytokines like IL-22, promote repair of damaged epithelial cells within that skin. But nevertheless, we still have no understanding what is this, how do these commensal bacteria educate the immune system to discriminate between what is beneficial and what is harmful. So when you have a activation of the immune response as a result of invasion or penetration of the skin barrier, it is what you've already learned about in the basics of immunology. So basically, the invasion of bacteria or damage to epithelial cells as a result of physical or chemical damage releases dams and pumps. This activates as a result of engagement of PRRs, will activate NF-kappa B in dendritic cells or macrophages, stimulating them to produce your inflammatory cytokines IL-1, IL-16, alpha, activate the expression of class 2 MHC, which will increase antigen presentation, activate chemokine receptor expression, allowing these cells to migrate to the nearest draining lymph nodes, and then present antigen to the circulating naive T cells to induce them to be effector T cells, but importantly, to cause these effector T cells to express this chemokine receptor, cutaneous leukocyte antigen, which will then cause those activated effector T cells to move out and directly home to those parts of the skin where that antigen is. And these effector T cells, depending on the antigen, can be Th1, Th2, or Th17 cells. After the infection has been dealt with, most of these activated T cells will die off because they've already performed their function, but a lot of them will then differentiate further into these memory T cells, which remember are mainly CD8, which then reside within this underlying tissue, such that on further infection, they can immediately be activated and kill off any virus infected cells. So much for these immune cells. Let's talk about some of the diseases. So psoriasis is a relatively common condition from sufferers, and it's essentially a type 4 mediated hypersensitivity reaction. So this is the result of mainly Th17 and Th1 responses. We don't know what the trigger is, but we think as a result of maybe too much sunlight, whatever, you're getting the necrosis of normal healthy cells, and that releases this particular antimicrobial peptide called cathelicidin LL37. What this antimicrobial peptide does is it combines with DNA And so once doing that, it forms a hapten carrier complex and a neoantigen, just like you learned in the autoimmune lecture, in the previous lecture. And by forming this hapten carrier complex, it gets taken up by antigen presenting cells, presents to T helper cells, which can then produce cytokines to induce this inflammatory response. As a result of the inflammatory response, you're getting your normal redness, swelling, but particularly itchiness. And this is a common feature of psoriasis, of this dry skin, this very itchy condition on that skin, all due to this inflammatory cytokine production. It's quite a debilitating disease with people who have serious psoriasis. So a lot of effort has been gone into trying to treat this condition. One of the more encouraging results as a result of this is through clinical trials using monoclonal antibodies to IL-17. So if you remember, IL-17 is an important cytokine for activating neutrophils. And so it's thought that by blocking IL-17 production, this has a major effect in suppressing the inflammatory response in this disease. Then pemphigus vulgaris is an example of the phenomenon of epitope spreading, which I told you about in the last lecture. So in your normal tissue with cell-to-cell contact through the adherents, basically what is happening is that in the initiation of this autoimmune disease, you have autoantibodies which are directed against the one epitope of these adherents. As the autoimmune disease progresses and this complete antigen is taken up and presented to T cells, the T cells produce cytokines which can then activate other B lymphocytes, making antibodies to other epitopes further downstream to what the original autoantibody was. And so with additional autoantibodies specific for other epitopes along these desmoglein molecules, so basically these antibodies then block the zippering of this adhesion molecule, preventing cells to stick to each other. And so you're getting the skin layers coming off because they're not joining with each other. And so you're getting this, this condition. And then finally, type 4 hypersensitivity, which, if you remember, is due to the activation of the cell-mediated immune response, particularly Th1 and Th17 cytokines produced by these cells. For your as medical workers or even laboratory workers, there are some people who have an allergy to latex. It means that they get severe rashes when they try to wear normal latex gloves, and so they have to wear special latex, non-latex gloves. What happens here is that the latex, being a small molecule, can penetrate the skin, and then the latex will then complex with normal cells or normal proteins in the body, normal molecules, forming a hapten carrier complex. This gets taken up by dendritic cells, gets presented to T cells, then activates those T cells into producing inflammatory cytokines, resulting in the redness, itchiness that you get as a result of this. A similar system occurs with many women who are allergic to jewelry because they're allergic to the metals or the metal ions in particular metals like copper or whatever. And similarly, the ions from this metal penetrate the skin, complex with normal cells, causes a hapten carrier complex, and then activates a cell-mediated immune response and the release of inflammatory cytokines. Okay, that's it.